Thank you, Ryan. Thank you, Fran. Thank you, SoilsWest. The people here today. It's a pleasure to be here. My name is Andrew Macrae. I've come from Federal University in Rio de Janeiro, and part of an exchange program. We're looking for collaborations. Today, I'm going to talk a little bit about some of the research I do, a little bit about UFRJ, and some future projects. So happy to, after take post questions. So the outline of my talk will be this. UFRJ first, about 10 minutes. Then I'll talk about postgraduate research in plant biotechnology and bioprocesses, which fits in with soils and agriculture. I'm going to give an example of some collaboration that was done with a neighbouring university and with people who moved on. And then a couple of future projects, which hopefully there could be some chances for us to collaborate. So first of all, UFRJ, that's a map of Brazil. And you can see that the state of Rio de Janeiro is relatively small and that our university has 4 campuses. Brazil, like Australia, is a large country. We're ever so slightly larger than Australia. I think we're 5th on the world list and Australia's 6th. I think our land mass is about 10% larger and we've got long coasts. I was just chatting earlier. Our latitudes are from about 30, I think it's 36 south to north. And Rio is right on the tropics at about 22 and a half. So just to give you an idea. The university I work at is old, bold. It's was the first university, and its history also extends back almost to when Australia's was colonized, shall we say. So we started off as an engineering area in 1792. Medicine about 200 years ago, then law. Like many traditional universities, they came together, and then all the other areas. So as a complete university, we're about 100 years old, and so share our mission, I think is quite important, is to contribute to scientific, technological, artistic, and cultural advancement of society through teaching, research, and extension. Research so that we can teach. So we're a research university. But the reason for research is then to share the knowledge, and then that spills into extension. And I think that's the same with this university. And we're public, and I'll talk a little bit more about that, share some of the culture of the Brazilian public universities, and the role then is to form a just, democratic, egalitarian society. So we have a political reason to be. Here's some pictures. That's Sugarloaf Mountain in the background there. You may have heard of Sugarloaf Mountain. Anyone here been to Brazil? I know you have. Anyone else? Yeah, so there's a couple. The rest of you should come. I think you'd like it. Yeah, please. It's a beautiful city, and I'm really lucky to have lived there for more than 20 years. And that's a picture of the old faculty building. That's where engineering is, and that's down in the bay next to the beaches. That's the old. There's some of our old buildings, original buildings from that time. So on the left, engineering, on the right, and the Faculty of Law. But where most of the university today is on an island that was constructed from 9 smaller islands out by the airport. So it was purpose-built, and that's where we are. Not nearly as nice as the old part of the university. We have about 100,000 people circulating on that island. Let's have a look at some of the details. So that's UFRJ on that island is right next to Petrobras, which is the state-owned oil company, and right next to its research center. So UFRJ is the oil and gas center for Brazil, and all of its research. So that large swimming pool-like structure, I think, is the deepest oceanic tank for engineering modeling. So that's on the campus, and then outdoors you have the usual other activities. With the oldest medical school, and during the pandemic, UFRJ played a critical role in providing all sorts of types of support in terms of testing and in terms of developing new ventilators. It took us a little while to adjust to the pandemic, quicker than some, and we had political challenges, but the governor of Sao Paulo decided that we would make our own vaccines and did it and delivered it, which was rather impressive from my point of view, and shared it as well. So Ultimately, given the difficulties of a country of 220 million people, and a lot of them living on top of each other, I think we did okay. Although the statistics might look different, I think with what was possible, we did okay. So we have, I think, about 9 hospital complexes in all, very large one, and we serve staff, students, and everyone else. I want to talk a little bit about teaching and research. I'm trying to be strict myself, 10 minutes for each area. I want to talk about this because it's a bit different. And if you've got someone that's come all the way over from Brazil, I'm hopefully going to explain some things which aren't quite the same. Let's go. So in our university, big, 53,000 undergraduate students. Nearly all of our students are on campus during the day. We have very few students studying at night, and we don't really do online. So they're all there. But what we do differently, I think, because we're public, is that we don't charge fees to study. If you manage to get a place at our university, then you're not paying fees. And what we did do 10 years ago is create policies to change the recruitment process. We created a national entrance exam rather than our own exam. Our own exam essentially meant that all of our students came from UFRJ, and they all came from the top 10 private schools. They were the ones that are best prepared to get in. There was a change for the federal universities. The federal universities created national exams, so anyone could have a chance, essentially. And we created a quota system that guaranteed that 50% of the spaces would go to people who've been previously excluded. There was a hoo-ha about that, saying, well, they'll never be able to cope, they'll fail, they'll drop out, and all the rest of it. And they won't have the minimum conditions to be able to come and study all day. And we did have problems, and we did have more dropouts, but what we've discovered over the last 10 years is that the schools have improved now that they can compete. The students have improved, and actually it's been a success. Our postgraduate research and productivity hasn't dropped, and that's where our students mainly come from. So I would say it's been a quota system has worked. What we had to change was we had to provide food. So we have university restaurants that charge about 50 Australian cents for a full meal, available to all students if you're prepared to stand in the queue. And you have to be, what that means in practical terms, I'll tell you all our secrets, we shouldn't really. That means that the lectures, people leave lectures about 11:30. So if you're teaching from the 10 till 12 slot, it's unwise to go much beyond 11:30 because people start disappearing off of the lunchtime queue. So there's had to be adaptations, but it works. And I often do straw polls to find out who does what. And for the vast majority of our students, that's their main meal of the day. So we've changed the demographic. We haven't changed the academic quality. It's taken a while, but I would say it's been a success. Right. Graduate students, not as many, 15,000, still quite a lot. We divide research into research groups, research areas, that then get funded from our Ministry of Education, which is called MET. And there's UFRJ, I think it's an interesting statistic, produces 10% of Brazil's research, impact research, which is a significant amount, given that we've got more than 100 universities in Brazil. So this is how we evaluate research. This is slightly different. We work in research groups, and every 4 years we do an evaluation process, which is based on impact, based on teachings, based on success of the students. So there's a number of criteria, and then you get a grade from 3 to 7. Essentially, if you're a 6 or a 7, then you're internationally interested. 6, you're internationally interesting. 7, then you're excellent. So you can see that's UFRJ. So we've got a mixture. It's not easy to get into the 6s and 7s. It's highly competitive. But once you get in, depending on those grades, that will impact the number of grants you can have and funding. These, for those of you who are interested, are the areas which are rated 7 at UFRJ. So we've got engineering, physics, geography, maths, microbiology, biophysics, medicine, genetics, morphology, biology, engineering, different types of engineering, medicine again, communication, sociology and anthropology. These groups are on a par with the best research groups in the world. In fact, in that, we get the whole. Oh, it doesn't go back, does it? Sorry.
The button below.
Let's go back. Sorry about that, guys. Okay. Whereas our output, looking at these graphs here, is that we have about 1,500 master students graduating every year and about 800 PhDs. So we're always looking for positions for postdocs. Anybody as a postdoc needs a good researcher, I can find you one. We also are heavily involved in extension and outreach, right? The kind of work that you do. It's a compulsory part of education at UFRJ. And we encourage and involve undergraduate students from the beginning by giving them grants. So we have undergraduate student grants for your sort of initial research projects for about 2,000 undergraduates. This means that more or less every academic who wants one can have their own undergraduate research intern who's being paid to do research. This then removes sometimes the argument that some areas are favored and others aren't. We managed to support all academics with one undergraduate student. And then we have a process of evaluating projects and meetings and celebrating that. Likewise, with outreach, we take the university's work and put it out into schools, into society, and we also support that with about another 1,000 grants. That money comes from the federal government, from the state government, from the university, from industry. 3/4 of it comes from outside of the university, 1/4 of it comes internally from the university money. So that's highly supported. And we, once a year, have a very large event saying, know us. So I'm not sure if Murdoch does that, but invites everyone in to know you. Right, that was UFRJ in about 12 minutes. Now I'd like to talk about plant biotechnology and bioprocesses. I was the coordinator of a postgraduate research program with this name. We are a group of about 20 researchers, a mixture of soil scientists, a mixture of plant scientists, a mixture of botanists. We've got people from sort of the pharmacy region, and we're looking in this group for research that will get us to a product. And it's kind of a prerequisite. If you're going to do a PhD in this group, right from the beginning, you have to have identified some kind of product that will go to industry. Program there. We work on this principle, which is the NIH One Health. All connected, healthy environment, healthy people, healthy planet, and biodiversity. So we're looking, biotechnology is all that biodiversity, and when you look at that graphic, the classic RNA, all biodiversity graphic, nearly all of that's bacteria. When you look at it like this, you can understand the argument there's bacteria, archaea, and we are archaea. This is the eukaryotes here, that's us. Okay, that little bit there. And we look into the biodiversity for biotechnological solutions. Now I'd like to talk about some projects that the group are involved in, which could lead to collaboration, and that I've been directly involved in. So one area that interests us is microbially enhanced ecosystem restoration, plant growth, carbon sequestration, pollution prevention, and mitigation. The importance of riparian buffer zones, the importance of trees in protecting environments and fixing carbon. And on the bottom of that slide is a couple of patents that our students do. So one of the ways that we measure ourselves is to encourage people to get to the point where they might ask for a patent, either an inoculant, some kind of probiotic, or some kind of kit detecting pollution or whatever. So we encourage that, and we reward academics and students for having that vision in the way that we evaluate them. Then probiotic and CRISPR climate change plants. Plant geneticists are interested in this, mixing microbes with plants to get better crops, essentially. And I think that falls into the SoilsWest area directly, previously as well with soils. Then water, protecting water. And in my case specifically, we're looking at different types of filters that we can enhance to protect natural waters. So that can be industrial, agricultural as well. And then this is more related to the oil and gas industry, but has applications elsewhere in human health, detecting microbial contaminants, real time with biosensors. So the fourth area that we've been interested in. Okay, so that was a quick overview of the postgraduate program in plant biotechnology, and now kind of why we're here, international collaboration. We understand that the more we collaborate, the larger the impact it has. And as I believe we're all working together with this idea of one planet, one health, and sustainability in one form or another. The fact that I can be here is a privilege, and to exchange ideas with you. So thank you for that. So here we have international government increases impact and outreach locally and globally, and want to exchange knowledge, exchange our best practices and learn yours. And the case study I put on here, kind of for fun, because it's coming to Australia, and most of you hadn't been to Brazil, because I thought I'd better take you on a fishing trip up the Amazon. So let's have a look at this. So in the middle of that picture is Professor Andy Whiteley. Can you see him? He's there on the right-hand side. This is the barbecue area just outside the Institute of Microbiology where I work. And he gave a course on basic bioinformatics and taxonomy. And one of the students there, Shayla, this young lady here, she was investigating and doing bioprospection for bacteriocins, looking at their use in food industries, as protecting food from souring, I guess. And she had discovered an awful lot of Aeromonas on fish. So, and from that we thought, well, that's interesting. We didn't know. We knew that Aeromonas were on fish. We didn't know they were on healthy fish, unhealthy fish. We didn't know that they were so antagonistic. We didn't know that they maybe could be useful in that kind of role. So we decided to go and test a few ideas and test that observation. She published those findings recently, as we were, doing some quite in-depth genomics and analysis. So that was the background to why. So lots of Antigus Aeromonas strains are from fish caught in a city lagoon. Fish are sources of diverse bacteria and bioactive genes. That's the question. Are they? Yes. Let's find out. So we come with these very simple questions. Can we use fish to biopress for genes and strains of specific biotechnological purposes? And can we use fish microbiome biodiversity to measure river water biodiversity? A couple of questions. The only way to answer that is to go fishing. That was the experimental design. We used 3 different fish species. One fish species that is all over the river, travels in shoals of fish at the same time, and that's called the piranha. It's a fish that will explore all the water in the river. We use another fish species, which is called the cachorra, which lives in the rapids. It's also known as I think the devilfish or the fangfish. You'll see a picture. And then finally, a catfish species, and they're the ones that lurk around the bottom, picking up whatever might be on the bottom. That's just there. This was the river. This is on the sort of southern edge of the Amazon rainforest here. Below here has been turned into soya. That's more or less the southern edge there, but there's still a lot of forest left. So I'm pleased to report there's an awful lot of Amazon rainforest left. A lot. So here, this is the river. We had friends in the river as well as the piranhas and the cachorras. There's some pretty big alligators there, so not advisable to go swimming. This is the red-eyed piranha, or also known as the black piranha. And this was one of the species which we were sampling. And this is the second fish, which is the jau. That's Professor Andrew Whiteley carefully taking a small specimen of that fish because they grow to be as big as me. That is the vampire fish, or cachorra. They're the fast swimming fish. So we sampled from their mouths and gill area. We sampled from their scales and skin, and we sampled from their anus, which would be, we understood, being the end of the gastrointestinal tract. Could be an interesting environment for bioprospecting. There's Professor Wiley filtering some water, and we've sequenced the DNA from that. Sampling, swabbing piranha teeth. You have to be a little bit careful when you're doing that. So some data. Hold on, let me get back. So there's the piranha there. What did we discover? So far, this was done just before the pandemic. The pandemic came, we all got locked down, samples got put in the freezer, and we all stopped. Those samples were taken out of the freezer a couple of months ago, and we've sampled 5 river water samples, and the first samples we've done just to see, out of interest, were the piranha anuses, just to see what might come out of those. And these are the data. Essentially, we got about 100,000 sequences from every sample, and from the swabs from the piranha, the vast majority, not surprisingly, bacteria and identifiable. So 95% identifiable. From the river water, still most of it bacteria, but other stuff, but only half of it identifiable. So half of the DNA we've got, we couldn't actually match it up with anything, which I'm always curious about. I said, well, what's that then? We haven't got the databases yet to know what it is, but we'll get there. Here we can see graphs, but what we found, which I think is interesting, is that in the river water there was like human DNA and monkey DNA, and I guess that must be sewage. But in one of the samples, it had 1% human DNA, and I wasn't really expecting that when there were so many other things that wasn't there. You know, there seemed to be more human DNA than fish DNA in the water sample, which struck me. Anyway, here are the dominant. It wasn't, in terms of the dominant genera, some really stood out. So from the fish, Cocuria, Staphylococcus, Enterococcus, and in the water, Mycobacterium, Mycolicibacterium, and that distribution there. These are just first glimpses, just for fun. We haven't done the detailed analysis yet. We haven't done all the other fish samples yet. But we're beginning to see things. The question, sorry, this, keep pressing on. What is missing from this PowerPoint is the slide which showed that we did find the bacteriocins. So we found the bacteriocins in all of the samples, apart from one, and we found many more in the fish tract. We didn't know that we were going to find them there, and I do believe that when we get the other data back from the fish surfaces, we will find Aeromonas, and we will find other typical fish genera which were missing, which aren't in the intestine and weren't detectable in the rivers. Next. So that was an idea of a bit of bioprospecting and collaboration. If you want to come and teach with us, I'm sure we can find an interesting experiment to do with you, and we can exchange some ideas. That's the idea. Some future projects that we will be doing, and this is one that has attracted some interest. It's a mangrove restoration project. It's sponsored by Shell with significant funding. Initially $5 million for this project. It'll be a large project and involve at least 20, team of 20, I would suggest, by the time we put it all together. This is an area of mangrove near Rio. It's an area that's been polluted next to an oil refinery, so we'll be doing some of the oil refinery work as well. And it's an area that we're going to restore. We have about 10 hectares, and we're going to plant about 16,000 trees there. Mangroves themselves, their importance, physical barrier, their biodiversity, their carbon sequestration, coastal protection, and fresh air, economic. There's an awful lot of value to a healthy mangrove forest. In Brazil, we have estimates for this. So in Brazil, the estimates are that 338 municipalities benefit from mangroves because we've got a very long coastline and a lot of it has mangrove. The value of that mangrove in Brazil is estimated at 5 billion. The value of mangroves, or the destruction or lost value of mangroves globally, has been estimated in Brazil to be about 40 billion. And so there's value there. And in terms of Brazil, 80% of the fish species of value, fishing species, are found or related to mangroves. So we have fish, we have crabs. There's an awful lot of value with mangroves. The objectives and the innovations of that project are listed here. So we want to be unique and the reference in terms of establishing a robust carbon credit certification for mangroves. And what we hope to learn from SoilsWest is some of the terrestrial side of that. So we'll be looking in the beginnings of a partnership here at carbon cycles. We'll also, because of the way that we replant mangrove and that we use plastic normally to replant mangrove trees, we put them in a plastic Coca-Cola bottle chopped off, and we do that because if not, the crabs will eat the young trees. So we need to protect the trees to a certain size from crabs. And the way that has been done has been plastic bottles, but we obviously don't want to use all that plastic. So we're hoping to come up with a bioplastic solution in partnership with some of the research that's going on here. There's some uniqueness there. So the innovations will be probiotic technology, possibly drones for planting in areas where people can't plant, but where we can involve local communities and local people, we will be, because many of the degraded mangrove areas are areas of extreme poverty, and the people that are living there are the lowest social profile, or possibly some of the poorest people in Brazil. They hunt crabs, and in 2 days they might find 8 crabs, and those 8 crabs might be worth about, let's put this on 30, about 4 or 5 Australian dollars. So they'll work for 2 days to earn 4 or 5 Australian dollars. There's extreme poverty, and it's destructive. The mangroves are also chopped down for firewood, and what we hope to instill is working with local communities, a system where we can restore in a sustainable way and return back to local communities some kind of wealth to make it in their interest to maintain the mangroves. So in terms of the scientific innovations, we'll be doing some eDNA stuff and some of the biodiversity stuff that we've seen with the fish. And we have got this sort of inclusion through education and training, and hopefully what we hope to produce is the how-to guide, which I think SoilsWest have done for some of the soil carbon. We like, this is an objective, how to manage in a more sustainable way your mangrove. And we know that it will depend in each community. We know that there isn't one size fits all, but hopefully we can identify the pathway so that we can restore and manage in a sustainable way these forests. So one of the things is no catch. You leave areas for a while so that they come back. The other thing, same with forestry. Yes, you can chop down forest if you want to, but in a sustainable way. A mangrove in Rio will grow to adult size in about 10 years. It doesn't take long for things to grow. These are the areas, that's Rio de Janeiro, and there's a big bay there that was mistaken as the River of January, Rio de Janeiro. It's a big bay. You can see there, and that's where we'll be harvesting from, and that's where we'll be collecting samples from. The university is that start there. Also have mangrove. This is on the far left-hand side. Here we have about 6,000 hectares of mangrove that has at various stages of restoration, and from that we hope to gain enough carbon credits to manage the rest of the project. We are told there are about 700 tons of carbon per hectare of mangrove, and that's something that needs to be measured and certified. So Vinicius will be doing will be leading the carbon work. We'll be looking at all the fluxes there. Biomass 2, we're doing old-fashioned measurements around trunks, but also hopefully modern measurements with LIDAR and infrared. Certification, I think this is the key. It hasn't been done for mangroves in Brazil, so we hopefully will establish that. Plant physiology involved with bioremediation, the probiotics, testing whether or not the microbial solutions really work. These are the species. So I've seen, last week I was in Brisbane and I saw a lot of mangue vermelho, red mangrove, which is Rhizophora mangue. Got plenty there. We've got mangue preto, black mangrove, which is Avicennia, and then we've got white mangrove, which is Laguncularia. I believe these are all here in Australia as well. They look like it when I was up in Brisbane. I don't know why black mangrove is called black mangrove and white mangrove is called white mangrove, because they're neither black nor white. They're definitely green. And the red one, okay, maybe because the seed pods look kind of red. But anyway, that's what they're called. So, probiotics, that'll be the area that I'm going to focus on, and maybe some of these new technologies, possibly with drones. But as I said, the key to sustainable reforestation and management is to involve local communities. So this would only be in areas where we can't do that. It's a picture of my lab. It's a little lab there with I've done quite a lot of work on isolating interesting bacteria that do interesting things, whether it's cleaning up pollution or helping a plant to grow. And one of my genuses that I like is bacillus, because they're particularly easy to grow and store and put in a product and sit on a shelf for a very long time and still work. So that's one of the areas that interest me there. The citizen science will come in. That involves Fabio and Laisa. They've been involved in a number of United Nations projects. They kind of understand it on a South American scale, but also the human side, which I believe will be key to the long-term success. It's the community work. They'll be managing that. And that's kind of the way we're going to be doing it. The socioeconomic baselining, it's talking to people, it's identifying within communities the champions, then supporting them, training them, educating them, and creating a source of income that maintains that while we carry on doing the rest of it. And that's the kind of work that we're doing. That seems to plot into Portuguese. So planting and managing, establishing the value of the forest. So creating value and understanding it. And then we're going to have guardians of the mangi, not guardians of the galaxy. We're going to have guardians of the mangi's mangrove. Happens to me. I think I'm speaking in English and then I'm speaking in Portuguese. So I apologize for that. Just happens. And data and publications. That'll be their symbol. There will be warrior crabs protecting our mangrove. And that's that project. It's large and we believe it'll repeat. Shell have asked, it's the intention that if we can create the model, that we then replicate the model along the coastline. So this is hopefully the beginning of something that will run and run and run, and we'll find ways of involving other people. Hopefully, it would be really nice to get Australia involved. We can. Other future project that I wanted to touch on briefly, which is directly SoilsWest. I had the opportunity to visit some farms in the state of Piranha, which is to the south of Rio, large agricultural areas, rich, good soils that have been essentially growing soya and corn. So there's a soya, corn, soya, corn, 2 or 3, I think it's 3 crops a year. They're very efficient. Everything grows very quickly and lovely looking soils when you look at them. You'd be jealous, I'm sure, because I imagine that your soils are yellow and orange and kind of dry and sandy. But I went along and had a look, and I met an agricultural consultancy company called Prime Agro, essentially a beef juicing company, but have now got into integrated farm management, and they understand the value of better soils. Even within an environment where you've got good soils, better soils is better. And they want us to add in to the soil quality index something about the biology and biological activity in an attempt to convince farmers that they should switch. Part of the argument for switching. And in this photo, what you can see is a multi-crop mixture of flowers and leguminous plants on the left-hand side, which is a break crop. So instead of corn, soya, soya, it's soya, corn, this. A mixture of eight or nine different plants grown at different rates, and then you bring in your cattle and they eat it, and it's controlled with an electric fence, and they eat and do their business all in the same place and carry on. And I thought it'd be more complicated, but having seen it's not too difficult. You don't need many farm workers to do it. And then have a look at the soils, and I was amazed, genuinely amazed. So on this photo here on the left-hand side is a soil under maize or corn, and there's just no roots in it. It's nice soil, texture-wise, a nice colour, but there's organic matter in it, but no roots. And then you go across, literally over there, 10 meters over there, to the other bit of soil on the farm on the other side of a road that has adopted this management for 7 years. And what you see is about 10 to 15 centimeters of rhizosphere under this type of vegetation. And even when you go, after that has been removed and the animals have done what they've had to do, and then you plant it again and you look underneath corn that's been through a rotation, you have about 10 centimeters of root-rich soil. And it's so rich. And the crop production goes up, and then they're producing healthy beef. And so essentially it's much more profitable farming action. The typical arable farmers have resistance to animals, but once they also see that their arable crops are improving, there's a hope. And what we've been asked to do is look at the biological function, not only the biodiversity, but look at what genes in there might be explaining it. So I think we'll be quantifying, not only saying what the DNA is characterizing, but we'll probably be quantifying as well. And finally, I mean, I've raced through, how are we doing for time? Are we all right for time?
Yeah.
Okay, so, getting this wrong. I believe there's potential as well for us to look at some of this stuff and work in collaboration with the SoilsWest group, coming up with a universal or Southern Hemisphere quality index that works looking at soil biological function based on the genetic data. I don't know, so something that we're looking at, not only who's there, but what other genes are there, and I think we need to quantify them as well, and adding that in to a soil quality index. So I apologize if I've raced a little bit, but hopefully that means there's time for questions. These are, I showed you 4 areas of research that the lab have been involved in, and these are the PhD students that have been involved in those projects. So Alana is a chemical engineer. She hasn't finished her PhD. She got pulled away to be a process engineer in the development of vaccines, and she's still doing it. Hopefully she'll come back and finish off her PhD, but she was working on the probiotic filters and biodegradation of dyes. So that's Alana. Next is Barbara. She's, in theory, going to defend her PhD thesis in December, and she's been looking at the bioinoculants and bacillus with me, and I'm pressurizing, let's get finished. Then there's Erica. And I didn't even speak about Erica. Erica is a coral probiotic person who has moved. She's actually defended her PhD. Is now in Kaust in Saudi Arabia, working on coral restoration. So the kind of microbial technologies go to other environments. There's Shayla, who's bacteriocins and fish. And then there's Rafaela on the right-hand side, who's also soon to finish, who's doing the immunosensors. So I'm here saying, need a good postdoc? All of them are good. I'm going to keep 2 of them, but I'm not going to tell you which 2. But there's 3 others that could be very useful in other projects. And that's it. Thank you very much for listening to my story, and happy to take any questions.